Good afternoon. Is it? Yeah, it's working. Good afternoon. Welcome. Welcome to the post-lunch session. It's session 3.7 on germplasm seed movement and global plant health. So this is a very important topic. There are a number of discussions even ahead of this starting official meeting about the seed phytosanitation, the various issues concerning the policies, the diagnostic tools that are being used, the implication of the new knowledge that is coming and how they are impacting the global distribution of germplasm, etc. So in this session, we have 6 talks touching different aspects, and 2 of them are from CGIAR to give some perspectives on how CG centers are organizing themselves in global distribution of germplasm, because as you know, many of you are aware, CG plays a very big role in making germplasm available for agriculture and agriculture research for development. We start the presentation. We have 3 invited speakers and other contributed presentations. One presenter could not make it, so we have, fortunately he sent his recording, so we will play it. Since I'm going to speak as one of the topic here, I let my colleague and co-chair, Dr. Safaa, to lead the session organization. Over to you, Safaa.
Good afternoon. Hope you enjoy your lunch today. It was good. Yeah. As Dr. Lava, my name is Safaa KUMARI. I'm from ICARDA. I'm handling the responsibility of the seed health and virology activity at ICARDA. I will help my colleagues, Lava. Lava is from other sister centers from IITA. As he mentioned, we have 6 speakers. The first speaker is invited, Dr. Yilmaz BALCI. He sent it to me a few lines about his biography. Dr. Yilmaz BALCI, he is the Assistant Director for the Planting Policy Program in USDA, Office Plant Protection and Quarantine. His team of 14 develops important policy for all propagative plant material imported into the United States, and consists of risk managers and policy specialists. His team issue federal important order to address the risk posed by emerging pests, as well as oversight of policy implementation at port of entry and offshore facilities. Dr. Yilmaz, you have 20 minutes, please.
How do this happen? Just click on the presentation.
Yeah, you should just click on your presentation, yeah. Let's move there.
Good afternoon, everyone. So the food was good. I hope you didn't eat too much. Otherwise, so it will be very easy presentation for me, probably, if you have eaten too much. Thank you again for the invite. And today, what I want to talk to you is about seed health. And I will be presenting in behalf of my team. Nathan Miller and Ed Potlakis work in my staff, but Ed retired and Nathan took a different job. So I ended up having this honor to come over and do some fun work here. And Tracy is our export specialist, and Nancy Osterbauer, our director for import regulations and manuals. So I work for APHIS, Animal and Plant Health Inspection Service, which is the NPPO for the United States. So what we do is we do issue a lot of the import requirements for anything that comes into the US for either propagation or for consumption. So the focus today is propagation and particularly seed. And to get started, first think about the seed trade, what is happening and how the seed trade is overall. And as this image here demonstrates, the seed is a pretty much global trade. So in one country, the breeder line is produced, the parental line, then it moves to the second country where you have the production of the foundation seed, and then it moves again to a third country for treatment and processing. It then still continues to move to another country for production of hybrid seed. It moves again to a treatment manufacture of commercial hybrid. It moves one more time, commercial packaging, and then final destination. So what it shows is 7 countries here, and it can go up to 12 countries. So a seed is, in that sense, became or has been to a certain extent a global endeavor. And when you think about all this movement, now you need to meet all these countries' import requirement. So you can start to think about how complex it can get. So the producer or whoever handles, whichever country handles it, they need to meet the import requirement. And it is stacked and stacked. And what happens if a new pathogen emerges in a particular country? So the linkage breaks. And then all of a sudden, you will not be able to ship it to that country for this next step. But on the other hand, the farmer is still waiting for the seed. So it's a huge disruption if a particular pathogen or new emerging organisms are identified. And throughout the meeting, we have seen that this is happening quite a bit. Either the global trade or climate change, or we as a human mess up things really well if we have the opportunity. So seed. In general, for many, many years, the import requirements were pretty straightforward, very simple. This is kind of generalization, but a phytosanitary certificate and a port of entry inspection. That's it. So seed will come in, and pretty much it will satisfy the import requirement. But what has changed? As I mentioned, new things start to emerge. In 2013, the CGMMV kind of emerged, which we issued a federal import order. And in 2018, the postbiviroids emerged, about 5 postbiviroids. Again, we had to issue a federal import order. And the same year, again, another virus, tomato brown rugose fruit virus, again, a federal order. And the impact was significant, of course. And I think one thing that you need to really think about it is, as an NPPO, what are your tools? You know, when these things emerge, one of the role APHIS has is safeguard. So we have to safeguard the country, the agriculture, natural resources for all these pests and pathogens. But at the same time, one of our role is to facilitate the trade, because it's import and export. At the end of the day, we need this, and that's helpful for us and for the other countries. So these federal orders, when we issue, we think about a lot of things, but I will get back to that same point later on, and what the challenges we have been facing. So to counter this emerging issues, we came up, we and collaborators, industry, academia, a number of people, with this idea of REFRESH. So REFRESH stands for Regulatory Framework for Seed Health. It's an acronym. In USDA, we like those acronyms. There's about 5,000 of them. So what it is systems approach that leverage industry best practices. So whatever is out in the industry, we want to use that in a systems approach and build on that. And then what we do is accreditation alternative to consignment inspection. So we would accredit a facility as instead of a consignment by consignment inspection. So, and what is the company or plant or the participating countries will do is 2 things: the quality management system and seed health management plan in general. So they have to develop that. So I'm going to talk about this, of course, a little more. But I want to take one step back, because I keep hearing this from different colleagues and countries, that we have to set the nomenclature straight. Sometimes what we say is not the same thing what the other country or what someone else understands it. Now, what is a systems approach, and what is kind of the traditional approach of managing these pests of concern in a phytosanitary term? So right now, let's say the traditional approach is reactive. So you find a pest, you have to do something about it. You take action of it. But in the systems approach, the idea is being proactive. So you think about the potential of a pest being emerging in a system. In a traditional approach, at the port of entry, we detect contaminants. So we target of something that is a contaminant or a hijacking insect or whatever. And in a systems approach, the idea is, to begin with, we want to prevent those contaminants. We just don't want to police, but we want to make sure that the system produces clean material. So there's a prevention there. And in a traditional approach, again, what we do is we produce and test. We produce of the material, plant material, and we test for the target things. But in a systems approach, the idea is to correct unsafe practices. So we don't need to keep testing, keep checking our system. So it's the practice that we are more worried about in the systems approach. And in the traditional approach, again, what we do is when an issue emerges, we do an endpoint inspection. So we fly or we go there and, okay, we try to find out why this particular thing has came up or emerged, why that contamination emerged. But in a systems approach, it's audit. So you look at the whole system, why did it happen, and what has led to this occurrence. So there is actually a fundamental shift from idea that you have from a traditional approach to a systems approach. As plant pathologists, most of you probably like this. So we want to produce clean material by looking at all sorts of different things. And that's really the foundational perspective in this refresh idea. So how does it work? Systems approach in a seed settings. What we are thinking is, first, the industry existed for hundreds of years. We are shipping seeds or moving seeds around for thousands of years probably. But let's say for the last several decades, the industry has developed so many things, tools, that they already utilize, which we could, as a regulator, take advantage of it, because they would actually target or be useful tools. And so what we want to do is we want to leverage seed industry best practices, whatever they are, and there may be a lot of differences. And second thing is, we want to focus on critical control points. So, and again, as a plant pathologist, it will be very simple for you to understand that. We want to look at when seed contamination can happen, when that pathogen can infest the material, and develop a mitigation to manage that. So, and then what we do is, as part of that, also that these practices are auditable. And at the end, a cumulative effect for pest risk reduction. So there will be critical points, multiple of them, and then in each point you do something independently. If one fails, you have the other one. If that fails, you have something else. You have other prohibitions potentially. So we want to focus on critical control points, and then the last thing is quarantine pests. So we want to make sure that the quarantine pests are being addressed in that whole system. So that's how the umbrella pretty much looks like. And how do we design the system, a phytosanitary HACCP system basically in this case, and this is not new in the food sector. It's being used extensively. Hazard analysis, critical control point system. And you start with first identifying the critical control points, whatever they are. And then you find out mitigation, specific mitigations that will work if on that critical control point. And then you would determine what are the methods and tools that you think it's okay, you accept it, it's working well. So you establish those performance standards, and then you need to establish also monitoring procedures to make sure that this is indeed working. If things get wrong, you need to establish corrective measures, who is doing what and how it's being done. And then at the end, you want to have a good record system that if NPP or someone asks, you can go throughout the whole production system and find out where things get wrong. So that's HACCP. And we are using this to develop our critical control points. And in total, for the seed pathway, there's 8 critical control points. It starts with site selection, or if it's produced in a greenhouse or in the field, whether that area is a pest-free area, pest-free place of production, or the site selection has to be in a place so distance from a particular quarantine organism. Of course, the seed, what you start, it has to be clean of or free of certain viruses, the soil, the material that you use. There is another critical control point. Next is the pre-harvest. So once it starts to grow, there are a number of things that you can do. You can spray, timing, inspecting, cooling, and so on. And things get contaminated during the pre-harvest stage. Once the seed is ready during the seed harvest, that's another critical control point for us. You can prescribe certain measures. Seed is harvested, it goes to the facility, either it's conditioned or there is some sort of a treatment. For example, there is standard post-behavior treatments in a number of the facilities. So you want to have a full control of that, how they use it, and what the treatment methodology is, and so on. And then, of course, once that is done, it moves to storage, handling, packaging type area, and there, again, there shouldn't be any mix, contamination, and so on. Oops. And of course, most companies have built in the seed quality testing aspect, where they check the purity and the quality of their seed, or whether it's free of certain things. So they will test it again. And at the end, there's the transportation part. So, and in all of these critical control points, something can happen. You want to have a good coverage on all these areas. So in the risk world, we look at the risk and try to reduce it in different stages. And in this, basically, what I'm trying to show here is, at the very beginning, the risk may be high, but gradually, as you move throughout the process, at the very end, the risk should be very low. And that's our goal, basically, in risk systems approach and risk reduction practices. And in each stages, you have tools available for you. And a tool that is acceptable, a tool that is maybe utilized by the company, a tool that you can negotiate. So how could all this work? Let's say US, we are producing cucumber seed, and then we export this using a phytosanitary certificate based on accreditation of the facility to Mexico, as an example. And Mexico accepts the phytosanitary certificate based on the accreditation that they also agree. And they will take it in lieu of inspection. So they will not necessarily inspect everything one by one, although they can do that. And then Mexico ships it to Australia, but Australia will accept the re-export phytosanitary certificate based on the accreditation data. and that they have accepted the facilities. So, and how did we support this, and how did we design this? So here's the refresh resources. It's an effort in making for several years. The COVID have, of course, had an impact on us, the way how we kind of moved this project. But the first version of it have been released for stakeholder comments. We received stakeholder comments, and this version 2 will be available on September 2023 on our webpage, and the last bracket there. And so the idea is here, first we have a concept paper which we describe how all this can work. Then we have the refresh accreditation standard. So this is the plan for the game. So who should do what and what we are looking for. And then we have also a support document for participants. So if a facility wants to participate, what do they need to supply? And then providing them resources in terms of the systems approach, what they can use. We also have a dedicated web page for this. So I'm going to go through these very quickly on accept the concept paper, what all this kind of include. First of all, the accreditation standard. Here we have information about the application and the enrollment. So what it's being needed and how an entity can enroll. The next thing is about actually the roles of the exporting and importing country. So as an exporting country or importing, what are your responsibilities? What do you need to do? Then the second thing is about the entity who is participating. Let's say Bayer as an example. So what they need to provide you. So the seed health management plan. So all this past viruses, whatever, how do they manage it in their practice? So they provide the information, identification, tracking, audit system, and reporting. All of that information must be provided so for us to evaluate participation. And then, of course, another important thing is what happens and who should do what if non-conformances are encountered, and what are the corrective measures. So basically, we are starting from the very beginning to the end to collect all the information that will be useful for us as an NPPO to understand the practice and tell and be clear of the roles and responsibilities. Exporting NPPO. What are the responsibilities of an exporting NPPO? Well, first, when they receive the application, they will have to approve or deny. They must maintain a refresh program. Currently, I am hiring additional program managers, so we are committed to have a staff at the NPPO who can address, who can follow, who will be the interacting and looking at the performance of the program. And then auditing part. So initially you start the accreditation, so full system audit, and then you will have the annual surveillance, partial audit, and then year 3, the re-accreditation full system audit. So it's based on audit. So basically we go and check and see whether everything is still okay as it has been documented in the agreement. So, and then suspending, reinstating, and based on non-compliances. So the participant manual, the most important thing is the seed health management plan. So they have to tell all the details about how they manage it. So how will be this operationally doable? So the entity applies to participate in a refresh and is registered and approved. Then you will have regular audits of refresh entities. So that will be the internal audits and by the company as well as by the NPPO. Then we have the port of entry documents where refresh certification will be provided and then seed released to the entity. And we still have the random inspection possibilities. What are we doing now? We need to put it in practice. So we have a pilot ongoing. With Chile, we are exchanging corn seed and high-value corn seed. And we are basically going through the whole system, how we can do this in a real world. So 2 companies submitted refresh manuals for review for APHIS. We reviewed, Chile reviewed, and we exchanged feedback. We provided the feedback to companies, and we have, for example, identified a particular virus that is endemic to US, but it's a concern for Chile, and how we're gonna do it under the refresh idea. And then I want to just touch on one other thing, which is NSHAP, which is, again, an acronym for National Seed Health Accreditation Pilot Program. So we started this in 2016, but the idea is it's based on the National Seed Health System, where we, companies participate through an agreement, and then they are being approved, whatever test that they can use. And so one consignment comes in, they will check it and self-report. So here in this case, the target virus is cucumber green mottle mosaic virus. And the NSHAP regulatory work is kind of based on consignment agreement, compliance agreement, and then a method that is being approved and results reported. Of course, no presentation is complete unless you have a bar chart, as we have seen in this meeting. So I have to have at least one. So here it is. And this shows that self-reporting, and the companies basically utilizing their own lab to test for cucumber mottle mosaic virus. And then when they find it, they report it and they destroy it. We cover all that under the compliance agreement. So in summary, Refresh is a long-term PPQ initiative to enhance phytosanitary protection in the seed pathway. So it's our long-term vision that as an alternative to the existing system, it relies on cooperation between multiple governments, academia, or seed industry partners. So all basically have a role to play, and that collaboration really makes it successful. And it recognizes best practices to facilitate global seed trade. And here are our partners, SAC, the Chilean NPPO, both Chilean and the US seed trade associations have played an important role, and the 2 companies who are right now participating are Bayer and Corteva. With that, I can take any questions. Thank you.
Thank you so much for the interesting presentation. Okay, we have time for 1 or 2 questions. Okay, but we need a microphone, please.
Yeah.
Can we start from the lady here?
Thank you so much. I'm from India, National Bureau of Plant Genetic Resources. And thank you very much. We do get the germplasm from USA. However, recently, USDA says we cannot give the declaration the consignment is free from ABCD, whether it is a virus or a fungus or bacteria, or whether it is a watermelon or maize. It has become a norm now for germplasm. So what is your opinion?
Well,
why is it so? Like, we think USA is such an advanced country with so much of facilities, but still to give germplasm, USDA these days are saying you waive off the additional declarations.
yeah, I mean, that's certainly a different question. I may provide you more information. I don't know the details of, depends on the germplasm, on the seed, and what the main questions are. But when we have certain import requirements, it's really pest-based, and unless it's like a property material that we don't have the right, so of course we will not do anything on that, but the germplasm facility itself is the oversight. We as NPPO more or less are out of that, except that we tell what the import requirements are or the permit conditions, if matter of fact that is the case. But maybe we can talk, and so I understand what exactly it is, so I can provide you more feedback.
Okay, okay. Any other questions? Okay, thank you so much.
Yeah.
Thanks for coming, thanks for presentation. The second speaker, Dr. Lava Kumar, is the head of the Germplasm Healthy Unit and Virology and Diagnostic at the IITA in Nigeria. As he is the leader of the CGIAR Germplasm Healthy Component, he is focused on ensuring the safe international transfer of germplasm and preventing the transboundary spread of pests and pathogens. He has initiated the green pass concept for fast-tracking international distribution of clean germplasm for food and agriculture research. And I'm sure he will talk about this in his presentation. You have 15 minutes, Dr. Lava.
Yeah. Thank you, Safaa. Yeah, thanks for introducing the topic already. And I would like to thank my previous speaker who set the scene for the systems approach. So I deliberately slotted my talk following yours so that I don't have to again provide a background to what is systems approach. In this presentation, I basically touch upon three elements. First, the significant role that CG centers play with regard to the enabling partners' access to important germplasm for various purposes, including breeding or direct use for agriculture purposes. And then secondly, how we are managing phytosanitary aspects. And that's where the role of the germplasm health units comes in. Very briefly, I won't go into details. I pretty much will keep referring to the previous presentation on some of the steps. And lastly, I deal with some of the challenges that we encounter with regard to germplasm distribution and a solution. When I say germplasm distribution, I mainly talk from the small sample research perspective. Much of the previous presentation talked about the commercial exports, big consignments. When you distribute germplasm from gene banks or breeding programs, quite often the sample size is very small, and that itself poses a different challenge when it comes to regulatory testing. So how we can tackle that? So I represent a CG system. I'm coming from IITA. All my colleagues, as listed in the authors, they are represent 11 centers, all of which are involved in germplasm enhancement. CG does various things. One of it is concerning with the safeguarding germplasm, which are nearly 700,000 plus accessions in its gene banks. And they are often used in the breeding programs and other genetic enhancement activities. So gene banks often considered as crown jewels of CG system. We have 11 gene banks spread across different parts of the world. What you see on the map at the headquarters is, but there are also substations and other in situ conservation centers, altogether 37 locations from which, where the germplasm is conserved, and they're used, all these stations are used for distribution purposes as well. We have quite a lot of diverse varieties. You can see it's a large collection. 93% of it is represented as seed, and 3.3% as in vitro collection, because we also deal with clonally propagated crops. And then we have field banks, mainly concerned with the trees. So altogether representing 35 genera and more than 1,000 species. This is super complicated when you think of phytosanitary management. So what you see on this map, that every green dot and the purple dot, these are all the locations from which the germplasm has been acquired over the last 50 years. And from there, the material have been moved, and then they are conserved in the CG gene banks. This is pretty much a global resource, what we are hosting. How does this germplasm flows happen? Just as a simple example, just like the commercial seed example that was shown in the previous presentation, most often the collections are made from different countries, and they are moved into the gene bank location, and there is a process involved. So from centers of production to the gene banks, and there is a process involved with regeneration, phytosanitation, and then regeneration for seed bulking and the distribution. And these materials are further distributed for the CG programs as well as for the partners programs for further use, either breeding, or trait evaluation, or more recently for genotyping purposes. And then from the conservation centers, they are also widely distributed upon demand, on request. Anybody can go to the CG website, they can readily see the list, and they can place order. And these materials are distributed under SMTA, Standard Material Transfer Agreement. And we also have another unique activity called safety duplication, wherein we package material and we ship exclusively for conservation purpose, safekeeping. They are not used in the country where they have been shipped to. And the only time that the box will be reopened is when it's repatriated to the parent location. So this is again a very unique chain where the material will go just for storage purpose and then it will come back for reuse in the same country. It is not going to be reopened. So each of these pathway has different phytosanitary demands. And these things are happening. So just as an example, this is not just theory, this is a very active things. In 2022, you can see the numbers of accessions that are being moving. The number that you see for conservation, this is the material that is regenerated for bulking purpose, so that the material is tested, make sure that the material is free from pathogens, pests, and then put back into the gene bank and make it ready for distribution upon demand. And then there are exports and the imports. This is where we do the surveillance monitoring. You just pay attention to the percent contamination. This is where we remove the material. So we do not remove the material that can be treated or salvageable. So that means this is the material that goes out because most often we cannot treat them. So in 2022, we have exported almost to 112 countries from the 11 centers. So if you have to map like this, it looks like a kind of an airline map. It's quite busy. Yeah, you can imagine the question asked, like in terms of declarations and all, how much work we had to do. And the recipients are different types of users. Majority of them are in the national programs. And this work is very well acknowledged, as in one of the editorials mentioned. CG gene banks play a pivotal role and in helping the global agriculture, especially dealing with climate change, the nutritional, improving the nutrition and the food content, etc. Just one example, how this germplasm distribution actually helping tackle the cassava mosaic disease outbreak that happened in East Asia. So cassava mosaic is present both in Asia and Africa. The species are different. The Asian strains species are different from those occurring in Africa. When the outbreak happened in East Asia, these are the outbreak caused by the Asian isolates moving into some of the East Asian countries. There was a severe reduction in yield. It's almost started wiping out the cassava production. Fortunately, the resistant varieties are available in Africa because this is again an endemic problem here. So these resistant varieties have been previously tested and known to be resistant for other Asian species. So upon demand, the varieties which are suitable for Asian climate were identified and shipped, and they have been put into practice. They have been tested and also started breeding program, and they're doing some of the varieties which are already going and ready for release. So this is an impact, just as one example. There are many examples like this. But then seed distribution comes with its own challenges. Yes, it is very important to have access to quality genetic resources, but at the same time, there is also a risk that these activities can accidentally contribute to spread of pests and pathogens, especially those which are seed-borne, because seed is known pathway for pest distribution, either as a source, as a host, or as a contaminant. Sometimes it serves as a passive carrier. It is indeed a risk, well-recognized risk, and it is a threat both to agriculture as well as biodiversity. And moreover, as you can see, in many countries where we are operating, the phytosanitary capacities are weak. That means if you make any mistake in preparing germplasm, and it's very likely that it can be introduced into the environment and then cause severe damage. So therefore, we need to be very careful when we move this material and make sure that the material is perfectly clean. And how we do that, and that's where the germplasm health units come. And this is something, a risk that has been realized at the beginning of the CG system establishment itself, because CG has been moving germplasm. They realized there is a potential risk as we move material. The CG system has to take care, not just only rely on quarantine agencies, but it is important the system put in place, considering the various weaknesses across different countries, that you would not take any chance when the material is distributed. So in a typical sense, germplasm health units work as a kind of internal quarantine organizations. So any material that goes out or comes into the center, they have to pass through the germplasm health units. And they use a number of procedures to make sure that the material is free from pests and pathogens. And we have been using what we call systems approach right from almost from the beginning, because the emphasis all the time is on producing clean germplasm, using the rigorous procedures, considering the diagnostic tools, the pest knowledge available on that day for the given pest and diseases. So we have different protocols. Some of them have, it's an evolving field, so we continuously update these things. And we have internal quality management systems. Just one example, as a clonal crop, it goes through a rigorous system of generating virus-free planting material, testing involving the most sophisticated technologies that provide sensitive detection, and we can identify, we do both targeted testing as well as broad specific detection to make sure that the germplasm is indeed free from all known viruses and before they get shipped. And similarly for seed crops, they are often regenerated under contained conditions to protect from vector-borne diseases. And we do various process. Once the seed is harvested, it go for further testing before they are conserved in the gene banks. And when they are shipping, before shipping, we also do mandatory treatments that includes fumigation and seed treatment subject to the conditions of the importing countries. Considering that we are dealing with a lot of crop species, so we rely on different diagnostic tools, both target specific as well as generic tools, looking at even unknowns. So we do both. In this process, we focus on both conventional methods as well as most modern methods such as high throughput sequencing. In this process, we also focus on developing our own procedures. As we identify new pathogens, we also develop our own tools. There is one specific example in this session, so you would hear some of these things. So even if I'm not giving details, we have another example from the CG system. You can get a better idea. One thing that we need to understand here is we apply all this in the context of the guidelines provided by the National Phytosanitary Organizations. This is very important. We can't just use any diagnostic, we can't just use any phytosanitary method. It has to be in line with the guidelines provided. So this is again a very important consideration that we take into. And then we also have a good system with regard to making decisions. Okay, most often we receive phytosanitary permits which are very generic. So how do we make decisions with regard to the distribution of germplasm? We have very complicated matrix, what we call decision matrix. We basically do not provide material for planting purpose unless it is free from pest. That is our simple motto. But there are a lot of dilemmas. How do we navigate germplasm? There are a lot of issues. The first being genomic-based surveillance. This is a bit like this, Hubble and the James Webb Space Telescope. High throughput sequencing has completely changed the game. The resolution of the data that knowledge it can provide is terrific. It's quite good. But then the challenge is for quite a number of things that we are finding, we don't have the biological relevance, which is very critical for pest risk assessments. And we are also finding a lot of viruses or other kind of pathogens in asymptomatic plants, cryptic. How do we handle these cryptic pathogens? So they are a big challenge when it comes to making decisions with regard to distributions. So considering the germplasm movement is all policy dependencies, so the new knowledge is posing a big challenge, both in terms of decision making and also how this is factored in the pest list of the national programs. So we have ISPM 38, it provides some leeway, but there are still a lot of gaps. From germplasm exchange point of view, from the CG side, we have variable demands coming from the phytosanitary agencies. And these procedures are continuously evolving. Sometimes the requirements are ambiguous. It is difficult to comprehend why these particular things are being asked. And all of it ultimately causing the delays in the clearance and loss of viability in seed viability in the transit, sometimes also loss of crop season. At the same time, we also understand from where these questions are coming. Sometimes it is because the pest lists are not updated, doubts on the health status of source of origin, lack of trust on the declarations, delayed processing. So no matter what the situation is, sometimes we come up to the situation, we do all these things, sometimes we lose seed viability or shipments are lost in transit. What is the way forward? We need a system, something like health management regulations which are used for humans. So just as an analogy, many of people who traveled in Latin America or Africa, we travel with yellow fever certificate, approved by the World Health Organization. It is one standard system accepted by all phytosanitary human health testing agencies. And during COVID, we have seen the challenge with every country having their own regulations. Yeah? It's super complicated. What is healthy for one country is not healthy for another country. And we have tier 1 country, tier 2 country, tier 3 countries. And I'm using it example because we are all fresh. It is a similar situation is happening with regard to the plant declarations. What we need is something similar to yellow fever vaccination, standard approach, where every phytosanitary agency adopts a single system. How does that can happen? They need to have a trust, as mentioned in the previous presentation, audits, accreditation. Anybody traveling through US, they have a TSA separate line. If you are TSA accredited, you can just pass through. You don't have to go through the rigorous check. So we started thinking in a similar way, like since CG system is doing the systems approach with all the rigor in our production system, if there can be an accreditation to our gene banks or the distribution programs, then the material that is generated from CG centers can be shipped much faster because it is very important for international crop development programs. So that's where we have come up with the system called Green Pass, which is as an analogy, it is similar to TSA Pre. And we have now something called Refresh. So there is a room for these kind of programs because there are ISPMs from IPPC which provide some of these guidelines. The most recent one being ISPM 45, which enables NPPOs to authorize entities to perform phytosanitary actions. So it is possible. Like Refresh, like I'm just thinking, US and Chile is just one country. If US has to have this Refresh accepted by every country, that means you need to have this kind of bilateral agreements with every NPPO, which probably is very challenging. What if there is a kind of a global system coming from IPPC to support these kind of programs? That makes a big difference. So these are some of the questions that we want to discuss a bit more in detail in the roundtable, which is going to happen tomorrow. We need a global consultation. I took some liberty to put some logos here based on the discussions that were happening in the last 2 days. International Seed Federation, the USDA Refresh Program. I know this EPO, the Europe has a generic resource bridge where they're also thinking some of network approach for rapid distribution of germplasm. And this kind of consultation can be hosted by G4 as a global body to bring in all the national programs, et cetera. Prescribe the best methods to avoid delays in germplasm distribution. And what are the technical issues that are concerning pest risk assessment? What are how to process the new knowledge that is coming from these novel technologies? And there is a need for advocacy for standardized policy, which everybody can adopt. And then awareness on the benefits of such system for safe germplasm exchange. So please do join tomorrow. We'll discuss a bit more in detail. That is tomorrow at 16 to 18:30. With that, I conclude my talk, and I'm sure I exceeded my time. Safaa, you have not said 1 minute. Maybe the chair privilege, but yeah. So I pause here. Thank you very much.
Thanks, Lava, for very interesting presentation. Let's have 1 or 2 questions. Please.
Yeah, you mentioned some of the challenges where a lot of your germplasm is present in very low quantity. So how do you do an effective test when you're such a valuable amount of germplasm that you don't want to dissipate?
Yeah, that's a big challenge. We do test, for example, sometimes we only provide 5 seed. When there are some germplasm which are difficult to regenerate. So seed production rate is very low. So when somebody requests material, we just provide 5 seed or 5 tissue culture tubes. And NPPOs have a standard to take sample. If they use all the 5 seed for testing purposes, then there is nothing left for research. But there are some understandings where we operate, for example, in ICRISAT, which is another CG system, where they work closely with quarantine. They understand CG, we get good support from the quarantine organizations. What they do, they regenerate that seed, and they provide the seed harvested from that particular lot. So that means you need to wait for the full season. So that is one approach some quarantine agents are using. Yeah, that's an open-ended question. But we do say that we already tested, you don't have to retest, but NPPOs don't agree. Yeah. So thank you very much. So we can discuss further tomorrow some of these issues.
Any other question? Hello. For tomorrow. Okay, let's move for the other invited speakers. Dr. Karen. GARRETT is a professor in the Plant Pathology Department, Global Food System Institute, and Emerging Pathogen Institute at the University of Florida. Her work addresses epidemiology, ecology, system analysis for decision support, including machine learning approaches. She is on the editorial committee for the annual review of phytopathology, research senior editor for phytopathology, and elected AAAS, I don't know what does mean, fellow, and serves in the presidency sequence for the American Phytopathological Society as a current president-elect, and she will be president for 2024 to 2025. Congratulations. Please, you have 20 minutes.
Yeah, thank you. Can you hear me okay like this? Okay. Yeah, thank you very much for the opportunity to present today. So I'll be talking about some tools for the particular type of systems approach that we've been developing. Yeah, and so we're looking at the 2 sides of the coin that others have talked about, because we want 2 things to happen that might be in conflict, right? We want improved varieties to spread easily through the systems, and we want pathogens and pests not to spread through the systems, right? So that's a challenge to try to make both of these things happen simultaneously. And we have a lot of other challenges in the greater context, right? The increasing human population, climate change, extreme weather events, and then of course war and political instability, as we've heard stories in this conference that make it more difficult to achieve all the things we want from systems. And just to mention one vision for what could be happening globally, if we're thinking of these systems in the larger context, a global surveillance system, and then ideally linked with a global mitigation system for crop disease. So one thing that's interesting to me to think about too is then how can we get all countries involved as full partners in this sort of global system. Yeah, so the 3 topics I'll be hitting on then today are characterizing seed systems and some different approaches we use for that, this R2M rapid risk assessment to support mitigation toolbox that we've been working on, and then a little bit more about scenario analyses for regional management. So here's a meme I like. You know, we want no regrets adaptation, and the best way to do that is to improve our systems so that even if our predictions for what will happen under climate change or new trade structures, et cetera, are not correct, our systems will be able to adjust for the new scenarios that come up. So I'll be trying to address how to improve systems. Yeah, so one approach we take sometimes is a network model approach. So you could think about that, this is a systems approach compared to some of the other discussions that takes into account more explicitly the spatial structure of systems. So instead of just talking about like there's one type of stakeholder in the system, we're like splitting that up. Maybe this one type of stakeholder has 20 representatives in the system. So we're splitting that up so we can think about the roles that those different representatives of that type of stakeholder have. So a node might be that kind of stakeholder or a farm, something like that, and then the link is something in our context of seed, like movement between those farms or stakeholders. Can change over time, and we can think about the volume of interaction and so on. And once we start characterizing a network, we can see some of the traits right away, right? So if we think of this socioeconomic network of people or institutions or farms or exchange of planting material. So once we can characterize the network, we can start to see what makes some of the nodes important in that network, right? Like in a little one like this is kind of obvious. You don't necessarily have to do many analyses. You can see that toward the center, those nodes are going to be more important. Like if you only have so many resources to try to manage spread of a pathogen or the attempt to make an improved variety spread more, you can see that the center nodes here are probably more important by lots of these different kinds of criteria. But of course, once you have a bigger system, it's not necessarily something you can just look at visually and tease apart, so that's why more formal analyses can be helpful. And so then if we think about these global systems, like here are some versions of cassava, potato, rice, and wheat crop breeding networks. So here a node represents a breeding program, and a link represents sharing of plant material. And so you can think about these again from the two sides of the coin, right? It's great to have resistance genes with the opportunity to deploy them in a lot of different places, but as this material spreads around, it's a potential problem. And then in general, I think it's interesting to think about these systems in terms of how well the current systems, which have adapted with lots of different forces in place, how well they allow us to adapt to new challenges. And here, I'll show a figure in a moment that's from a paper that several great collaborators had worked on. So here we were looking at a system, like what is the system that we want to build in place globally? So something like this, and here I've put a red box around what we're focusing on here is like the movement of pathogen, or model, how pathogens disperse, or in the context of trade here, right? And so ideally, we'd like to have that embedded in this bigger system where we can look at all the different kinds of data layers for microbe presence and absence, disease effects, incorporate that into models, and then have that be part of a global understanding of how systems work and what the critical control points are where we can improve the systems for plant health. So just as an example of one type of trade network, here's for stored grain. And so this was a paper that looked at US and Australia, but I'll show you the rail trade network for wheat in the US, which captures a lot of the trade that's there. So as you can see here, so here the state nodes are larger if they have more wheat production, and the links are, the weights are indicated by thicker links where there's more trade. So if you understand this, you can start to put in place like a trace forward, trace back kind of system, right? So if you discover the spread of an invasive pest or pathogen or an important subpopulation like pesticide resistance populations, then you can start to figure out where it might have already spread by the time you detect it, or where it might have come from and where else it might have spread to. Yeah, and when we're looking at seed systems in other countries, here's an illustration from potato in Ecuador. So here we're thinking about both the potato transactions in this small snapshot of a seed system, and also the sources of information that people get for IPM, which could be one risk factor. So we can evaluate the system in terms of how good the sources of information are for people that are protecting the health of the potatoes in this case. So here's what this little system looks like, and we can start to divide the different players in the system into different categories, right? So there's an important source of seed for the system that we're evaluating. And then there are sort of like two levels of fairly important players in the system. And then there are the outskirts sort of the system as well that could be a source of a pathogen into the system, but that aren't characterized as well. So again, this is a little system, so you can just sort of look at it and pretty much imagine what's happening with it. But then as you start to look at bigger systems with more people, a more formal analysis can be useful too. And this is an example of using an epidemic simulation to determine how important different players in the system would be for monitoring. Yeah, and then talk a little bit about this R2M toolbox that we're working on. So the idea here is to come up with a set of tools for looking at epidemiology in plant systems that make the most of existing data and knowledge so that you could implement them quickly without needing to do a new field survey, though of course that could be a great addition for new data. So here we're looking at tools for expert knowledge elicitation, cropland connectivity analysis, scenario analysis, trade network analysis, and then we could think about adding other sorts of tools, also like climate-based risk is of course an important perspective when you have data about climate-based risk for particular pathogens. And so we were joking about how if we make these tools really easy to use, then they could become very common. Anytime you're studying a system, you could use this toolbox for a quick analysis. And with the idea of potential users like extensionist scientists, national plant protection organizations, and for thinking about collaboration with CGIAR, different sorts of CGIAR tools too that could be part of an open source ecosystem. And so thinking about the excuse me, expert knowledge elicitation, you know, everybody's talking about language models and what they can give us, how they can summarize data. But so it's interesting to think about like old-fashioned knowledge, right, that's in human brains and how we can make the most of that. So thinking about expert knowledge elicitation in that context. So the idea here is that sort of a systematic way for getting information about what people know, and here are some examples where we've worked with that, and how to effectively get information. And so the idea here that we can think of this expert knowledge in the Bayesian framework, if you like think about that statistics framework, was the prior knowledge about a system and then the posterior. So the prior knowledge, the expert knowledge could represent that prior knowledge that people have about a system, and then new experiments can build on that in terms of the posterior knowledge. And we're developing this as a meta tool in the sense that we're coming up with a set of curated questions that could be used that over time we hone to make them have a better quality, right? Because questions can be vague, people tend to misunderstand them, but over time you can understand how to make them better. So that's one type of information we can get. Here's an example from expert knowledge elicitation in Pakistan. We were looking at experts' estimates of crop loss, in this case to wheat leaf rust, and then also looking at, so there's not necessarily, well, maybe there's rarely a lot of information about informal trade, but experts often have a pretty good idea of what informal trade is like. So here's an illustration from this of looking at both formal and informal trade in this system. Yeah, and so I think for these sorts of methods, it's interesting to think about standard operating procedures and how we can make these more general tools for the systems. And so the scientist in me sometimes doesn't really like the idea of a standard operating procedure because we should always be coming up with new ideas and so on. But we can think of a system like this, right, where at some point people want standard operating procedures, but then there can be the individual and team creativity that's sort of feeding into that, and then where the standard operating procedures come up short, there can be like a feedback to, again, generate individual and team creativity to keep improving tools. So that's part of what we were thinking about, for example, with the expert knowledge elicitation tool, that you could keep incorporating people's good ideas about how to improve the questions as a community resource. We can also think about the value of information and cost of information. I think it's good to keep those in mind, like, is the cost of generating different kinds of information justified now? But that, of course, we can't always anticipate what will be useful in the future, so we don't want to cut ourselves short too there. So do we anticipate the cost could be justified in the future? If you're interested in looking at those kinds of concepts of value of information and cost of information, here's a paper that we applied some of those ideas in looking at management performance mapping. So how well does seed health management work for potato seed health in Kenya and Ecuador. And then finally, a moment about scenario analysis in this context for thinking about regional management of seed health. So the idea that if you can, well, a lot of good can come out from even just thinking about how to characterize the scenario, right? Once you try to characterize the scenario carefully, you realize what you know and don't know. So you might, just in the trying to start to implement it, you might learn about what you need to, what kind of data you need to collect. And then once you implement it, then it can give decision support, right, for management. So here's a framework that I've been thinking about a lot. So the idea that you could have the biophysical network like the lower level. So that's, for example, maybe the distribution of seed, maybe the associated distribution, unintentional distribution of pathogens with the seed. And then the top level would be the decision makers who are, in effect, determining how much of the spread can take place. And they're making decisions based on economic considerations, just what it appears that they need to do based on what other people do, and so on. And so the combination of those two layers can help to determine whether there are good or bad outcomes for the system, for example, in terms of epidemic spread. So we have an R package available to do that analysis, and we're in the process of making an online app for non-R users to characterize the system and then see what the outcomes would be for different sorts of management choices. Yeah, and so it also has tools for looking at the surveillance value of different nodes. So if you can characterize the network of spread for a seed system, for example, then you can use this tool to evaluate the network and see where the most important points are for surveillance and mitigation. And then in the more complete scenario analysis, looking at the social and biophysical parts of the network, you can use that scenario analysis to ask questions about like the most important locations, how benefits, if the nodes are people, you can characterize by gender and age, for example, how the benefits are distributed, who's at greater risk in that system. How could subsidies and policies influence system outcomes, and is the system working consistently with what is desired for it? One way to characterize a landscape, once you start thinking about spread within a landscape, either in terms of spread by vectors or windborne spores, for example, or in terms of local informal spread, then cropland connectivity can be a proxy for some of those processes. So that's one way to take a landscape and convert it into a network for consideration of some of these processes. We can think about that as sort of a first approximation. Like if we're developing more complete scenario analyses, cropland connectivity is a relatively simple, like first approach to approximating what's, of course, probably a more complicated system. So here's an example of an application of some of these types of tools in the Republic of Georgia by Kelsey Anderson Onofre and this team. And so here we were looking at characterizing the potato seed system in Georgia. And one of the threats there is Syncytium endobioticum, and including different subpopulations of Syncytium endobioticum. So we started looking at the results of an expert elicitation workshop and incorporating those in modeling. So we could look at simulated potential pathogen spread to understand the importance of different locations based on the information from experts, cropland connectivity to give a bigger, rapid assessment of the area, and then also looking at international trade risk analysis for that system. And then another example of applying the sort of impact network analysis, looking at the socioeconomic layer and the biophysical layer, is this paper about avocado systems looking at laurel wilt. And so I have to admit it's not so much a seed issue here as just a general epidemic issue, but it illustrates some of what people have to deal with at a regional scale. So in this paper, looking at the question, under which epidemic scenarios is regional management most successful? And using this sort of model I just showed of looking at socioeconomic layer, epidemic layer. And then this study incorporates a couple different policy scenarios, carrots or sticks, whether you're helping people by subsidizing their good management or whether you're penalizing people if they're not implementing good management. And it also includes some modeling of how people make these decisions, how stubborn are they versus how quickly do they change what they're doing with new sorts of information. So one of the interesting results there was that in fact, if at some points where it's expensive to manage disease and people can get away without managing disease, sometimes more information about how they can get away without managing disease can actually make the overall regional management more complicated. Yeah, and so I'd invite you, I'll show this slide again in a moment too, but Berea Etherton is going in that especially red box there, is going to be talking about some of these tools more, and then also this project with avocados too. So you could see her poster for more details about that model and how that system works. So as I'm wrapping up, as I'm wrapping up, I'll just mention that we're working to keep improving these R2M tools, expert knowledge elicitation, scenario analysis, et cetera, and thinking about how to scale them up for effective mitigation, and really interested in other input as we develop this open source ecosystem. And especially we're this fall working to develop the version 1.0 of these expert knowledge elicitation questions, our question catalog and analysis catalog. So if that's something that's really interesting to you to think about, please talk with me about that, because we want to include a lot of different perspectives on what kind of questions are important for that question catalog for supporting plant health. So I'd like to close by thanking some of our different funders for these projects who provided support both in terms of the funding for the projects and also a lot of intellectual input as well. And then if you'd like to see some of these others, so the red box shows the ones that are still coming up that you could still catch. So I think all of these illustrate some of these different tools and their applications. So I hope you'll give these a look as well. Thank you.
Exactly 20 minutes. Thank you so much.
Oh, so no, no time for questions?
Okay, please. We have 2 questions here.
Yeah, hi, really comprehensive, this good stuff. I didn't read the paper on the work in Kenya, so maybe that answers this question. You can tell me, Karen, to just read the paper. But when you're looking at that value of information versus the cost of information, how does it actually work out when the value of a specific information type might change from situation to situation? So for example, knowing that an organism occurs in another place, you may not know that in one case, you may know that in another, but so that would definitely change the value and the cost of getting that information. How do you apply that in the real world?
Oh, well, that's a very good point. So an analysis of the value of information and the cost is going to be specific to a moment, right? And so when there's an opportunity, it's valuable to keep updating it, right? So I guess like a lot of analyses, if there are limited resources, there may just be one analysis that becomes less valuable over time as things change, but ideally you could keep updating it with the new circumstances. Yeah.
So thank you for the talk. My main question is, there's been concerns recently about ChatGPT and its like water usage and its energy usage, and I'm wondering what your plans are for the future for like using these AI and making sure they don't like contribute to the climate crisis that you're trying to prevent the impact of, if that makes sense. Because I see that they're very useful tools, but obviously they need a lot of computational power. And I'm just wondering, sort of, are you building around that, I suppose, is what I'm asking.
Oh, okay. Yeah, that's a really interesting question that I haven't really thought about that much, because some of these analyses are pretty computationally intensive. So I guess that's another kind of cost of information, right? That I have to admit I haven't really figured out that well yet, but I should. I mean, that would be a more complete evaluation of the cost of the information.
Yeah. Thank you.
Thank you. Okay, Dr. Maka, can I say no for my boss?
Thank you. One of the early slides, you mentioned about global pest surveillance system. Is it something in place or it is something desired? I did not get the point.
Oh, let's see. Well, so this was a proposal for what could be put in place, and it's slowly developing. As you can imagine, it's a giant project. I think others can tell you more about that than I can, because CGIAR is continuing efforts in this. Well, maybe if you want to say something, Lon.
Yeah, there is some efforts going on in this CGIAR plant health initiative, especially by linking the germplasm health laboratories spreading across different world to start working with national plant production organizations, improving their capacity, etc. Yeah, it requires much massive engagement, but we started. Yeah.
Thank you so much. Thank you.
Thank you.
The last presentation for Dr. Celia Chalam from, she is a principal scientist and head of Division of Plant Quarantine, ICAR-National Board of Plant Genetic Resources from India. She has expertise in policy issues related to the plant biosecurity and biosafety of GMO. She has organized more than 30 training workshops for scientists, plant quarantine, and custom officially on biosecurity and biosafety. She is now current a member of the International Committee for Plant Virus Epidemiology of the International Society for Plant Pathology. You have 15 minutes. Good luck.
Thank you, Dr. Safaa. And the topic is, at the outset, I would like to thank the organizers and the scientific committee of ICPP2023 for giving me an opportunity to speak here. The topic is quarantine of germplasm for biosecurity against transboundary viruses, importance of diagnostics, and phytosanitary regulations. And this is my outline of my presentation. I'll give a brief introduction, although this gathering doesn't need an introduction, and then international, national regulatory framework, diagnostics, and what we do at NBPGR, Delhi, India. And it was already dealt with this, along with the transboundary movement of plant material, whether it is a commercial material or germplasm, both carry the inherent risk of introduction of pests. In my talk, when I say pest, it is fungi, bacteria, virus, nematodes, even the weeds also. And this example of transboundary spread of pests, like for example, banana bunchy top virus, which has come from Sri Lanka to India. Still we are not able to eradicate that, and we have a big program by Department of Biotechnology to give the virus-free and true-to-type planting material to the farmers. And the latest outbreak in Bangladesh in 2016, it is related to the series of grain imports from Brazil, led to the wheat blast outbreak in Bangladesh, and recently in African country also it has been reported. And what we need for plant biosecurity is regulations, diagnostics, and phytosanitary treatments to salvage the material, and the capacity development of both the scientists and the quarantine or biosecurity and customs officials. Why I'm saying regulations, if we have diagnostics and if it is not regulated, suppose I have a protocol to detect bean pod mottle virus, but India regulation doesn't say that we don't need bean pod mottle virus, it doesn't have a relevance. And if we say we don't want bean pod mottle virus, but if I don't have diagnostics, again it has no relevance. And these are some of the international standards for phytosanitary measures. Dr. Lava has already dealt with it. And for this international standards for phytosanitary measures, Indian government has national standards for phytosanitary measures. For the paucity of time, I'm not showing all of them, but for example, ISPM 34 is on post-entry quarantine stations, and India has the standard operating procedures for post-entry quarantine facilities. And also we have the diagnostic protocols for regulated pests, and we do have the framework for pest risk analysis. Depending on this, Indian government has Plant Quarantine Order 2003, and unlike USA, Australia, India has a two-tier system. The commercial material is dealt by Directorate of Plant Protection, Quarantine and Storage, and the germplasm is dealt by NBPGR, where I come from. And we have 73 plant quarantine stations which dealt with commercial material across the country, and germplasm is dealt by NBPGR at New Delhi, that is the capital city of India, and also Hyderabad is at the southern part of the country. And India is free from several pests. We have more than 1,200 pests are regulated as per the Plant Quarantine Order 2003, and out of which 264 are viruses. And also when the germplasm comes into the country, we do the pest risk analysis, like other speakers have already dealt with it. For us, Plant Quarantine Order 2003 is a Bible or Bhagavad Gita or Quran, and we do use crop protection compendium by CABI, and in one click we know what are the pests which can come along with the crop. And we also brought out edited volumes of potential quarantine pests for India, crop group wise, cereals, grain legumes, edible oil seeds, and the recent one is tropical and subtropical fruit crops. And this is the whole story of how we do the quarantine processing of exotic germplasm in India at NBPGR. And for the transgenics, we need the clearance from DBT, Government of India, and others, non-transgenics, we don't need that clearance. And then Director NBPGR gives the import permit depending upon the country specific. Suppose if it is from USA, we say that the germplasm should be free from soybean downy mildew, bean pod mottle virus, tomato ring spot virus, etc. And then we do once the material comes into the country, all the specialists, entomologists, nematologists, pathologists, virologists, weed scientists examine it, and then it goes for specialized detection. And then if there is anything which is infested and infected, we have the salvaging techniques, that is the phytosanitary treatments. And if we cannot salvage, then the material is rejected and incinerated. And apart from this, all the legume germplasm, if it is soybean, pea, cowpea, faba bean, all that black gram, green gram grown in post-entry quarantine greenhouses. And the transgenic germplasm is grown in containment facility, which is of BSL2 level, and then tested for pests and also the terminator gene. So the testing for GMOs is also part of quarantine in India, the germplasm. And this is where all the scientists look for the symptomatic seeds, and all the symptomatic seeds are removed. This is just an example of avocado from Australia. And all the weeds, soil, everything is removed, and then it goes for specialized tests. Like for insects, we have the X-ray radiography. The seeds looks absolutely healthy, but when you expose them to the X-ray, you can, I don't know whether you can see the dots here, they are the hidden infestation of bruchids and chalcids. And also we use the molecular techniques, and some of the interceptions are given here. And fungi and bacteria, we use both conventional and molecular techniques, including the multiplex PCR and loop-mediated isothermal amplification, and some of the interceptions I gave here. And the nematodes, similarly, we use both conventional and molecular techniques. And the weeds, we have a weed scientist, and some of the germplasm really comes with the weeds, and those weeds are taken out and incinerated. And coming to the virology part, all the symptomatic seeds, like here mottling, this can be due to soybean mosaic virus, which we already have in India, or bean pod mottle virus, not reported from India. And then this one is broad bean stain virus, not reported from India. And pea seed-borne mosaic virus, it is there in India, but pathotype 4 is not there. So all these symptomatic seeds are removed, and only healthy looking seeds will go to post-entry quarantine growing. And we use both soil-based greenhouse and then the benchtop greenhouse. And when you have the symptomatic ones, we isolate them with a muslin cloth bag. And we also have the provision to release the material to the indenter, and indenter will grow them in the post-entry quarantine facility, which is a certified facility by the designated inspection authorities. So this is just an example. And all the symptomatic and asymptomatic also, representative to 10% of the healthy looking also, we test them. and we do the electron microscopy, which gives us an idea about the shape and size and which group it belongs to. Depending on the pest risk, we do the ELISA, and we are fortunate to have the anti-sera for about 70 viruses. And once wherever the results are ambiguous, then we go for PCR, and most of the seed transmitted viruses are so far RNA viruses, so we do the reverse transcription PCR. These are some of the viruses: cherry leaf roll virus, tomato ring spot virus, raspberry ring spot virus, bean pod mottle virus. These are not reported from India. And also we have multiplex RT-PCR protocols. This is in case of soybean, 5 viruses simultaneously. And also we do have protocols for real-time RT-PCR, for example, bean pod mottle virus, and also helicase-dependent amplification, and LAMP, loop-mediated isothermal amplification. But don't think that we do all these techniques. We do only 2 techniques, and these are 19 viruses which are intercepted, and I'll just skip this. And also we have certain salvaging treatments, and we uproot and dispose following the standard operating procedure. And yeah, we do, we are R&D arm of the Directorate of Plant Protection, Quarantine and Storage. We help them to identify the pests. This is the case where Bromus cicalinus was abundant in US wheat fields, and then we have not imported that. And this is case of basil from Germany and then Niger from Ethiopia. We helped the DPPQs not to import the commercial material because of the seeds, and these are some of the challenges. As Dr. Lava mentioned, sample size is very small, and detecting an unknown virus is an issue. And then of course, anti-sera are expensive, and capacity building is, we do take care. And India also has a national plant we don't have the national plant diagnostic network per se, but we have a very excellent system for national certification system for tissue culture raised plants, where the system has referral labs and accredited labs, and the tissue culture plants which are true to type and virus free are given to the farmers. You can see 76 facilities are recognized and 5 accredited labs. And we do have the system in place, referral labs for detection of GMO or LMOs across the country in 4 places. And recently in 2023, India has inaugurated Clean Plant Program with a budget of $2.6 million US with diagnostic centers, clean centers, post-entry quarantine facilities, so that the disease-free planting material will be given to the farmers. And in future, this is only a proposal. We have been giving this proposal for the past 10 years. India would like to have the National Plant Pest Diagnostic Network in line with US and Australia. When we have the system in place for tissue culture or GMOs, we are hopeful one day India will have this. And we conduct the training programs, as Safaa has already mentioned, for both customs officials and plant quarantine officials also, apart from the scientists, because in India, the customs have the search and seize power, not the plant quarantine. So now 400 customs officials are trained in this, and I will not read all this. One thing is, usually I don't talk about it in formal meetings. As I have explained recently, the USA, all the germplasm, they ask us to give the waiver. When we give the waiver, some of the USDA scientists are writing to the Indian counterparts, you take the waiver from NBPGR. It has become so popular now. And because we give the waiver so that we can import and do the stringent quarantine, what we do, you have seen all this presentation, so that we get the germplasm for the crop improvement programs and do the stringent quarantine and release it to the scientists. And that's it. Of course, the regional working group for biosecurity, at least one should have, and the roster of experts. Thank you so much.
Thank you so much. Oh my God.
Any questions?
We have time for one question. Okay, thank you so much. Let's be careful. We have unfortunately, we have one speaker. He was not able to join us. He sent a record for his presentation. Dr. Rafi Keterbala. Dr. Rafi is the Executive Secretary of the Asian Pacific Association of the Agricultural Research Institution in Bangkok. He is executing a number of regional global projects, including projects on phytosanitary compliance for international seed movement, and also on pesticide risk mitigation, and also on One Health. Work for National Agricultural Resource System in India for 3 decades, mainly contributing towards plant quarantine and germplasm health. Served for CABI South Asia as regional director for 7 years, and also as consultant for 11 projects of FAO, UNDP, and World Bank on SPS WTO matter. Okay, well, let's hear about his presentation. It's exactly 20 minutes.
Greetings from Bangkok. We are in the very important session on germplasm, seed movement, and global plant health. And I'm grateful to the organizers for allowing me to present through this video my topic on public-private partnership model to enhance safe international seed trade in the global South, a case study of South and Southeast Asia. Before I come to the topic, let me thank Dr. Lava Kumar and Dr. Safaa Kumari from IITA and from ICARDA respectively, who have organized this session in a very nice way and who has ensured that there is a diverse kind of presentation in the session, to which I am not a beneficiary to, unfortunately, to listen to all the presentations. However, I would like to say, whatever I'm going to speak on seed trade in this presentation, in general, applies to germplasm health also, and germplasm movement also. It is seed, which is a commodity, and germplasm, which is a research material. So the global norms are a bit complex when we look at seed and germplasm together. But the WTO standards only pertain to international seed trade, and for germplasm, there are a lot of things to be done, which is being done, and by certain agencies which you all might have discussed before my presentation. Now, coming to the topic. This topic is a part of many research projects which APARI, where I'm working, is undertaking with the mission of strengthening agri-food research and innovation for sustainable agricultural development in Asia and the Pacific. Is under risk mitigation thematic area that we have projects related to phytosanitation and innovation from STDF of WTO, from USDA, and from FAO. And we have more than 80 members in APARI, which are worldwide, but mainly in the Asia-Pacific region. And among these members, we have 22 country members which have their national plant protection organizations, who are the key beneficiaries of the work I am going to speak on. Now, what is needed at a very basic level for addressing phytosanitary measures? We have realized that agriculture is increasingly involving complex interactions among stakeholders at multiple levels. Agriculture innovation needs a system perspective. When it needs a system perspective, phytosanitary measures can only be better addressed if we have a system perspective. And what is that system perspective? System perspective, if you look in the flow chart here on the left-hand side, you see clearly is a bridging of the research and education with the private enterprises through some bridging institutions in an enabling environment and completely supported by science and technology policy, by the political system, by the science actors, and by technology from other sectors. So you need a cocktail of the actors, the players to build a system on which we can build upon to achieve some specific goals and objectives. And this pertains to phytosanitary measures completely. Now, we are going to talk about seed movement. Let us look at the background of seed movement. I'm not going to only speak on the SPS measures, very important for compliances for seed trade, which is of course there when we work on the ground. That's our subject. But we need to appreciate the complexity of the matter. Look at the approvals we need from ministries and institutions when we do seed trade and germplasm movement. There are various technical factors which come into play even before we make the movement or trade. Research and development, very critical, then registration, then production, then certification. There are a lot of commercial aspects which are dealt by the companies at their own levels. And then there are compliances, various compliances at national and international level. On the left-hand side and the right-hand side, I have listed few agencies and organizations and treaties and all. If you read it carefully, you will know all what you need to know, all what you need to comply with when you do international trade or at times even the domestic seed trade and germplasm movement. There's a multidimensionality of requirements for seed trade and germplasm movement also. For seed trade specifically, you have pillars of environmental, plant health and food safety requirements which I have tried to highlight here, the 5 pillars. And when we talk of only the SPS agreement of WTO, it is the pillars of food safety and of plant and animal health which assume special importance. However, we cannot ignore in the comprehensiveness of the subject the product quality, the environmental factors, and certain social factors which play a role in these requirements. If you look at the forecasted value of global seed market, it's very interesting. Look at the quantum jump we have from the year 2017 to this year of 2023, where a value of 86 billion US dollar is being forecasted and is almost being achieved. What does it show? It shows increasing importance of non-tariff measures such as phytosanitary measures and how they are going to play a role. And even in a study, it was found that these non-tariff measures now have become much more important than the tariff measures in terms of the global trade. With all these points in view, I would like to say we have realized in APARI there was a dire need to build upon the outputs or successes of many global and regional projects which took place here, which includes projects from Asia Pacific Plant Protection Commission, from USDA, from FAO, and from STDF of WTO. And what we realized that there are some important phytosanitary concerns in Asia which needs a lot of focus. They have been talked about. In some cases, actions have been taken. and there is a diverse situation in the countries because of the diverse economies in the region, because of the diverse level of growth of countries in the region, and the diversity in the functioning of the NPPOs. So among them, the 5 which we have listed, which we took up for studies under SKDF, which is a SPS capacity working, which has a SPS capacity working group, and which funds or sponsors projects for improving the SPS capacity globally. We have undertaken the project based on the 5 parameters. Number 1, pest lists are not updated for seed transmitted viruses. This we have worked out with the NPPOs. Impact to the PRA process is long and not specific for seeds or for the seed transmitted pest. We are not having a digital phytosanitary certification system that can facilitate the movement of agriculture goods in the region. There's a lack of capacity to comply with international standards, that is ISPM 38, which pertains to international movement of seed. There's a lack of coordination of partnership of NPPOs with the private sector and which relates to the multinational seed companies from the private sector mainly. Having said that, the project is being piloted in 6 countries: Bangladesh, Cambodia, Laos, Nepal, Philippines, Thailand, and Vietnam. 7 countries, I'm sorry, and with the technical support from Asia Pacific Plant Protection Commission and from the NPPOs of Australia and New Zealand. And we have the 5 implementing partners. In fact, apart from APARI, we have 4: Asia Pacific Seed Association, CropLife Asia, International Seed Federation, and American Seed Trade Association. These partners were very important from the private sector to achieve the multiple targets of the project. There is a specific allocation of work for APARI, which is a project lead, and on the technical side, we 5 partners have assured who will be doing what and how. Now, the goal of the project is increasing seed trade and market access for the Asia Pacific region. And hence the objective is to ensure that phytosanitary issues do not impede the trade of seed between these countries. Phytosanitary capabilities match with global standards, and public-private trust and partnership is boosted, which is very crucial to ensure food security through availability of high-quality seeds. The key areas of engagement is identifying gaps between the infrastructure and capabilities of NPPOs, and this is the output 1, and the activities here have been achieved. I'll come to that at the end of my presentation. Then creation of a database where all information relating to important phytosanitary requirements for the countries in the region are recorded. Consolidation of the portal of regulated pests and import conditions for the region is another very important engagement. Then capacity building for PRA and other related ISPMs for export certification, international seed movement, requirements of the NPPOs, the pest risk analysis, surveillance, pest-free areas, and so on. Advocate the adoption of initiatives such as ePhyto. This ePhyto has become very crucial in this era of digitalization. Promoting the involvement of investment of the private sector, how much we can engage with the private sector by involving them, by engaging them through their support, through their laboratories, through their financial support, and how to facilitate the lab accreditation program for the independent private laboratories by the NPPOs as per the norms of ISPM 45. When you look at the E-phyto scenario alone, we have seen that out of 112 countries participating across the globe on the E-phyto hub, 45 countries are participating through the generic system, whereas we have an ASEAN window here, and more than 30 countries from the region are yet to be registered either on this central hub or the ASEAN hub. Now, another point is focusing on the technical plus functional capacities. Why I say that? Because generally only technical capacities are built, and efforts are not being done to ensure that we build the capacity to navigate complexity, capacity for collaboration, capacity for reflection and learning, and capacity to engage in strategic and political processes when we build the technical capacity, all of which actually are called soft skills, and they actually build or enhance the capacity to innovate. And above all, the capacity development, what we are seeking here is more in a more holistic manner. It is not the individual capacity alone, it's the capacity of the organization and of the enabling environment. So, as I mentioned, the project, what we have, has outputs and its activities. The output one, as I just mentioned, which was pertaining to country assessment of the gaps, has been achieved, so I'm not going to dwell much upon this. Then output 2, yes, we are working on it, and we aim to have a prototype of the whole system to develop a portal and a fully functional database of phytosanitary requirements, and database will be pertaining to the seed transmitted pest in the list of the pests of the NPPOs. We'll be strengthening NPPO's capacity for related to various ISPMs, such as ISPM2 on pest risk analysis, ISPM6, which pertains to surveillance, ISPM11, which again relates to pest risk analysis, ISPM 12, which talks of phytosanitary certificate issuance, and then ISPM 29, which relates to the activities of the pest-free areas, then ISPM 38, which relates to international movement of seeds, and finally ISPM 45, which relates to the requirements of the National Plant Protection Organizations for accrediting the laboratories of the private sector. So we have a couple of programs, and we would like to here in this output 3, building NPPO capacity, we would like to provide all assistance in implementation of the E-phyto for countries that use ASEAN new single window system. Output 4 is very crucial. We have all the private players with us as technical implementing partners, and with the support of them, we are creating a public-public and public-private coordination platform. We have joint workshops foreseen here to see how we do accreditation of the health testing laboratories of the private sector, and there'll be policy dialogue for developing a laboratory program of independent private laboratories by the NPPOs, so that what we impart here is taken up by the policymakers at the end of the day. So we have a clear knowledge management strategy and a defined impact pathway assessment, and we have achieved the activities under output 1 so far, and capacity building activities have been planned and initiated. If you look at the gap analysis, the output of the output 1 of the various outputs of the project, you will see database of seed pest list does not exist in any of these 7 countries, except Thailand, where we have for food crops with relation to seed pest. Capacity building for E-phyto that is ISPM 12, is required almost everywhere except Thailand, where things are quite advanced. There's a need for PRA training, which has been done in abundance over the past years, but every country which even has got experts, they find need for looking at the most recent scenario and to work with seed transmitted pest. And international standard ISPM 38 training, which pertains to international movement of seed, I think is needed almost everywhere, though there have been trainings in Thailand and Vietnam, and all have expressed desire to work more on this. Seed health testing capacity, that is accreditation of the seed health testing lab of third party, which is the private sector here. Here, right now, the level of understanding is very low, except in Thailand or maybe in Philippines. In general, this will be focused upon. And we will also look at the phytosanitary capacity evaluation report where it has been done, but no further roadmap or strategy has been done, or where it is required, barring Thailand again, where we will see how other elements can be picked up from there to supplement and complement in our project outputs. So what we are going to do is more collaboration between industry and NPPOs. NPPOs will be more actively engaged, continuous dialogue for developing the web portal related to seed transmitted pest, continuous capacity building, both technical and functional, of the participating countries, NPPOs, and they would be adopting new standards and methods such as e-phytosystem. This is what we are going to achieve in the project with the public-private partnership model that we have created. And to sum up, I would like to thank the organizers who have given me this opportunity, and I would like to thank all the APARI partners and donors. Specifically here, I would like to thank SKDF, which has come forward to help us to meet our goals of the project. Thank you.
Thank you so much. Unfortunately, we don't have question for the last presentation, but hope Dr. Rafi will join us tomorrow with the roundtable session. And if you have any question, you can ask him directly, or you can contact him by email. And hope he will join us tomorrow. I don't know, Lava, would like to add?
Yeah. Firstly, we'd like to thank all the speakers. It's a tremendous job. This is a complicated subject of bringing together science, policy, and regulations to address the practical issues. So we had very good submissions, and we had quite a bit of challenge to select topics for this oral presentations. And I think while doing that, we ensured a mix of different presentations that gives a kind of a good perspective with regard to the needs of regulatory system, where regulatory system needs to be strengthened, as well as the technical challenges that we face and the practical issues. I think the presentations, the 6 presentations bring in, at least to me, they have done a very good justification in exposing things. So we still have an opportunity tomorrow to further discuss and deep dive into some of the key issues during the roundtable session and ask ourselves some pertinent questions. Likewise, we also can prepare questions for the regulators where they need to adopt and change. So please do join us tomorrow at 4 o'clock, right? 4:30, I guess.
4:30.
Yeah, 4:30. We will send again a reminder. So we'll have the speakers of the session as panel members, and also some other people who will join as technical experts. And everybody have equal voice on that round table or round chairs, because I don't think we have round chairs. I mean, sufficient tables to sit around. But we are going to arrange our chairs in a kind of a circle. So I think it's going to be open discussion. It's going to be very interesting, and we look forward to meeting you again. Thank you.
Thank you.